Welcome back to Dr. Liu's VCE Biology. Today's topic is responding to disease challenges and strategies, as presented in the 2024 Victorian Certificate of Education, VCE, Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. Gout is a common and painful form of inflammatory arthritis caused by the accumulation of uric acid crystals in the joints, resulting in sudden, severe attacks of pain, swelling, and redness. Option C correctly states that mast cells produce signaling molecules like histamine, which then act on blood vessels to cause the heat and swelling of the inflammatory response. Students who selected option B likely recognized that inflammation is an innate immune response, which is true. However, option B is incorrect because urate crystals, while present in healthy individuals, only accumulate and trigger these inflammatory attacks during gout flares. This investigation explored a potential link between alcohol consumption and gout development. To ensure a diverse sample, non-identifying data on gender, age, and ethnicity were collected. Participants, diagnosed with gout by their doctors, anonymously completed surveys detailing their gout attack frequency and alcohol intake. This type of study is typically a correlational study or a case study. The data collected was primary, as researchers gathered it directly, rather than using secondary or anecdotal sources. Adhering to ethical standards, all demographic data remained non-identifying. It's important to note that participants self-rating their attack frequency isn't considered an ethical standard. There are two main strategies to gain immunity. Passive immunity relies on the injection of ready-made antibodies. Active immunity, on the other hand, involves injecting antigens to prompt the body to produce its antibodies. Additionally, antibodies can be transferred from a mother to her fetus across the placenta. Neither antigens nor antibodies are given orally to babies. Antigenic drift involves small, gradual changes in viral surface antigens, which can impact existing immunity. In contrast, antigenic shift means sudden, major changes, typically when different animal strains co-infect the same host, resulting in new surface antigens. Influenza A's wide host range is a result of antigenic shift, not its cause. This makes option C incorrect, as it mistakenly claims a wide host range induces a shift and confuses it with drift. No information is provided about influenza severity. Similarly, major antigenic changes in option D describe antigenic shift, not drift. Also, the diagram doesn't show influenza B infecting goats and chickens, so we can't identify a threat to them. Option B is correct because antigenic drift in influenza C could mean humans lack pre-existing immunity to a new substrain, necessitating public health measures to prevent an outbreak. Trastuzumab and pertuzumab are monoclonal antibodies vital for treating HER2-positive breast cancer. Both specifically target and attach to the HER2 receptor on cancer cells. However, they achieve this by binding to distinct sites on the receptor, meaning each possesses a unique variable region. Students were expected to include both antibodies in their answers, but a frequent error was incorrectly stating that trastuzumab's variable region bound to the cell membrane, while pertuzumab's did not. Monoclonal antibodies contribute to treating HER2-positive cancer in the following ways: block HER2 receptor. They attach to and block the HER2 receptor, cutting off growth signals and preventing cancer cell proliferation. Deliver targeted payload. They can transport chemotherapy drugs or radioactive isotopes specifically to cancer cells. Enhance specificity. By primarily affecting cancer cells, they help reduce adverse side effects on healthy tissues. Activate immune response. They can flag cancer cells for destruction by immune cells or directly stimulate the immune system's attack. Limit metastasis. They can help to immobilize cancer cells and reduce their ability to spread. Measles, a highly contagious viral infection, is transmitted via respiratory droplets from infected individuals. Reported cases reached their highest in 1994, followed by a rapid decline. This reduction is directly linked to vaccination, which enhances herd immunity by diminishing the susceptible host population, thereby restricting measles transmission and leading to a decrease in incidence. Measles is no longer endemic in Australia, though the Victorian Department of Health issued a health alert in January 2024 after 3 new cases were reported. These infections likely stemmed from an infected traveller or an unvaccinated individual. Publicly listing the locations visited by infected persons offers several important benefits. Risk mitigation. It allows people to avoid potentially contaminated areas. Early detection. It prompts those who have visited the locations to monitor for symptoms and seek testing. Containment. It facilitates self-isolation for individuals who suspect exposure. Promoting safeguards. It encourages the use of protective measures like masks. Boosting immunity. It can promote uptake of booster vaccinations. The emergence of new pathogens in human populations is driven by several interconnected factors. Primarily, increased interaction between humans and animals facilitates zoonosis, where pathogens jump species, often exacerbated by greater proximity to wildlife or the wildlife trade. Within this context, antigenic shift can lead to entirely new viral strains through major genetic reassortment from co-infection by different animal viruses. Additionally, global environmental and human activities play a significant role. Climate and environmental changes may expose humans to novel microbes, e.g., from melting ice caps, while increased exploration and international travel accelerate the encounter with and global dissemination of previously unknown pathogens. Even advancements in biotechnology carry a slight risk of accidental pathogen release. It's important to distinguish these from antigenic drift, which involves only minor changes to existing pathogens, and antibiotic resistance, which makes existing bacteria harder to treat, but doesn't create new pathogens. The Disease Challenges and Strategies section is worth 18 marks, 15% of the total 120 marks, and is divided into multiple choice and short answer questions. Multiple choice questions, 4 marks, questions 17 and 18. Gout focuses the immune response to gout-causing crystals, the roles of dendritic cells and neutrophils, and alcohol's contribution to the condition. Question 22. Immunity types covers different forms of immunity, such as passive immunity acquired via placental antibody transfer to a fetus. Question 33. Influenza discusses various influenza virus strains, antigenic drift and shift, and public health responses to new variants. Short answer questions, 14 marks, questions 6A and 6B. Monoclonal antibodies explain the use of monoclonal antibodies like trastuzumab and pertuzumab in treating HER2-positive breast cancer. Questions 7A and 7B. Measles includes data on Australian measles cases and discusses strategies to reduce them, such as vaccination and public health alerts. Question 7C. Emerging pathogens describes factors contributing to the emergence of new pathogens, including zoonosis, climate change, and increased international travel. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.